Today we are going to talk about nucleic acids in our start of chapter 3. So nucleic acids are polymers. And so far we've seen that carbohydrates are polymers, and we've seen that proteins are polymers, and now this third of the 4 macromolecules are also polymers. And they have units that are monomers, and those monomers are called nucleotides. So nucleic acids are polymers, but they're specialists for information storage and transfer. And so we're going to see how they work as we move forward. So the two big molecules that are involved with this category are DNA, deoxyribonucleic acid, or RNA, which is ribonucleic acid. Now, when we look at the monomer units, we said that a nucleotide is the monomer for these different nucleic acids. And all nucleotides are made up of a sugar. So a pentose sugar, remember that pentose means that it has 5 carbons. It's going to have a nitrogen, what's called a base. So we're going to see the nitrogen base, or sometimes called a nitrogenous base, and a functional group, the phosphate group. Now, if we're only looking at the sugar and the nitrogen base, sometimes the term nucleosides is used instead of nucleotides. But as soon as you put in that phosphate group, which you're seeing down here, so there's the base, there's the 5-carbon sugar, or the pentose sugar. Here's the nucleoside. Add the phosphate, and there you go. So here's our monomer unit right here. The bases themselves, so the ones we just said were bases, are going to be one of 2 types. If it's a single ring, and I'll show you that in a second, then it's called a pyrimidine. And if it has a double ring structure, it's called a purine. The sugars, so these are the components of the bases, these are the options. And the sugars, they're either going to be DNA, it will have a particular sugar, which is the deoxy meaning without some oxygen, deoxyribose, which is a name for sugar, and RNA has ribose. Let's take a look at those. So again, a base plus the sugar plus the ribose sugar of some kind is going to be a nucleoside. Add the phosphate. Now we have this monomer unit, the nucleotide. The pyrimidines, which have the single rings, there are 3 of them, but only 2 of them we'll see in DNA. So DNA will have the cytosine one right here, the C. It'll have the thymine. And it will also, so these types, these pyrimidines, those are the 2 that the DNA has. But if it's RNA, in RNA has cytosine too, but it has uracil. So thymine, we'll put an X through there. Purines have a double ring structure. So here you can see how they are structured. Remember, these are the nitrogen bases. And so you can see nitrogen throughout these molecules, all of them. So adenine is a purine, and guanine is a purine. The bonds that form from those condensation reactions, or the dehydration synthesis, are called so there's the phosphate in there, diester. And you've seen esters before, so phosphodiester linkages. Now it's telling us here that nucleic acids grow in the 5' to 3' direction, and we'll see what that really translates to coming up. So if I have a nucleotide, which means I have this pentose sugar, I have a base, there's the pyrimidine base because it has a single ring, and there's the phosphate on it. We can orient it like this. So here's the phosphate on the top, and it looks just like this. And this side of the sugar, if you look at it, this side of the sugar is where that 5th carbon is. So when we say 5', we're looking at this side of this monomer unit. And then the 3 side, we're looking at the same monomer. We're looking right here at the 3rd carbon in this ring. So the 3', 5' idea has to do with where the carbon is on this sugar structure. But why that's important is because then when you take one of these monomers and you put it together with another one, you're going to see that it's going to create this 5', 3', and then it's going to join to the next one, 5', 3'. So it orients in that general direction. And this is where the dehydration takes place, right here. Here's the OH group here, our hydroxyl on this. Here's the H, hydroxyl on this, sorry. And you can see that's where water comes out. And that right there is known as the phospho. It's joined to a phosphate. And this is the diester right here, bond. And that happens every single time we have a monomer forming joining this chain. So it's going from this direction to this direction. All right, here we go. Now, if you have a group of these that's about 20 monomers, we can call that oligo nucleotides. So this prefix on it is just telling us that it's got a fairly short chain. So in this, which is 20 is pretty short for these for nucleic acids. But RNA is much smaller than DNA, so with RNA that's not unusual. We'll see though that RNA and DNA in most cases are pretty big, and so we'll call them polynucleotides. Oligo meaning few, poly meaning many. And as you can see on there, they're the largest polymers in the world, or natural polymers in the world. I should stick with that. So here's how we would compare DNA and RNA. RNA is a nucleic acid. It has ribose sugar as a part of its monomer unit. It has these 4 possibilities for the base, and then they are single-stranded, and I'll show you what that looks like. DNA has deoxyribose sugar, has everything except for instead of uracil, it's got thymine, and it's going to be double-stranded. So when they come together, especially when we're looking at DNA, what we're going to see is that the information is within the patterns of these pairings of the bases. So adenine to thymine, cytosine to guanine, so A to T, C to G. These always pair together when it's going to be a double-stranded molecule. That's called complementary base pairing. Now, how those base groups are paired is not by phosphodiester bonds, but by hydrogen bonds. And hydrogen bonds, we'll see here in a minute, they're pretty strong in DNA, particular in RNA, but they're not quite as strong as covalent bonds. But the reason this is kind of important is because the hydrogen bonds can be separated a little bit more readily than without major effort, where covalent bonds would be harder. So I'll show you here ultimately what we mean. RNA is single-stranded, but will fold, and we're going to see how this folding occurs by certain base pairs coming together. So here's a strand of RNA. It's a single strand with each of these monomer units just attached to one another. So here we have these, in this case, it'd be very few of these nucleotides joined. And here we see the 5' end on this one is down here. So it's going this direction, 5', and the 3' end is up here at the top. So when we talk about the orientation of this molecule, we say this one, we can see it running 5' to 3' in this direction. And here you can see what happens then to those long strands. So this would be what we'd see here is RNA, but it's starting to fold in, and certain of those base pairs are getting attracted to one another, and they bond by hydrogen bonding together. In DNA, we get 2 long polynucleotides that join by their bases, but then they twist, and they form that thing we know of as a double helix. So here, this is a good picture of this. So in this DNA, what you see is, here's the 5' end of this long set of nucleotides that are all now bound together by these phosphodiester bonds. So we're seeing the 5' end down here and the 3' end up here on this side. But then go to this side and you see the 5' on the top and the 3' on the bottom. But the other thing you'll notice is that thymine, there's T, is joined always with adenine. So if there's a T on this side, there's going to be an A on this side. If there's an A on this side, I'm down here, there's going to be a T on this side. So you can see that the bases that have the single ring always join with another base that has a double ring. The G, which has the double, joins with the C, which has a single. Same thing down here. But also notice one other thing that's kind of interesting here. So when A and T join together, see those dots? Remember, those are the hydrogen bonds. A and T join together by 2 hydrogen bonds. That's binding these 2 strands together. Where the G and the C have 3. And that's going to have some significance later on. So this whole thing is going to continue. So this is a very short little picture of it. You'd see this continue. It'd be much, much longer, but you'd always see that joining of A's to T's by hydrogen bonds, G's to C's by hydrogen bonds, 3 of these, 2 of these. A lot of detail. And that's what it ends up looking like. And you can see up here they're showing this is where that 3' end ended up, and here's where the 5' end ended up. But all along the way, C, G, G, C, A, T, and the hydrogen bonds are right in here. The phosphodiester bonds are over here. They'd be the part that are kind of forming this sort of outer ribbon on this DNA molecule. This is something you'll hear a lot, but DNA carries the information. This is the code of life. This is our genetic code. And we find the DNA in the nucleus of a cell. And so DNA has some really important functions. It's involved with it gets replicated when a new cell is formed, and it has these genes. It contains the genes that pretty much code for all of what we are, all of us. And we'll see how that all works. But DNA is in the cells, and DNA is used to make RNA, and RNA is used to make proteins, polypeptides. And let's see here. I think I'm going to quit right there for this first intro. I think that's plenty of info to look at. And so look at the figures that I've posted and see if this helps, and we'll talk when we get back.